Dear laureates, dear colleagues, ladies and gentlemen. Martin Rodbell received his training as a biochemist at Johns Hopkins and the University of Washington. He became gradually more and more interested in the cellular regulation of lipid metabolism and particularly in the actions of insulin. Among other things, Dr. Rodbell, who worked at the National Institutes of Health, developed a method to prepare isolated fat cells. This preparation proved extremely useful, not only in lipid metabolism and insulin work, but fat cells became widely used in all kinds of signal transduction studies. In particular, some open questions that were raised by the work of Earl Sutherland and Ted Rawle could be and were attacked in Martin Rodbell's laboratory. For the final assault, Martin Rodbell worked with 3 postdocs who were also very skilled experimentalists, Lutz Birnbaumer, Stephen Paul, and Michiel Krantz. And with tremendous intellectual audacity, he predicted the existence of a GTP-dependent transducer of a pertinacious nature. He's a member of several societies, including the National Academy of Sciences, and he has received several awards, including the Gardner Award. Dr. Rodbell, we are anxiously awaiting your Nobel lecture, Signal Transduction, Evolution of an Idea.
Well, first of all, thanks very much, Bertil, for this very kind introduction. I'm here today not only to talk about myself, but also to introduce my colleague and friend, Al Gilman, and I hope that I'm able to go through my lecture rapidly enough so that he has enough time to talk. But I think you will see that he and I have complemented each other in our work all these years. And I'm really very grateful and very fortunate, in fact, that he and I are able to be together at this momentous occasion. Now, I thought I would present 2 slides to sort of illustrate the kind of guy I am, because they represent some sayings that I think adequately, for those who know me, express the way I approach science. The first slide is a statement, first one by Alfred North Whitehead, who is a well-known American philosopher, who says, "Seek simplicity and then distrust it." And the reason I put that there is because the living process is very complex. And we have a tendency, of course, to try to simplify, and that's necessary in science. But for me, it's always been necessary to think in broader terms and to try to extract as much complexity out of a system so that I can begin to see the panoply of possibilities. The second is really by Mark Twain, whose real name was Sam Clements, who made a statement saying, "There is something fascinating about science. One gets such wholesome returns of conjecture out of such a trifling investment of fact." And that has been my hallmark. I'm one who loves to see a little bit of information and then make big conjectures. Mark Twain was absolutely right. But for me, that was an important part of my life. It has made science so wonderful for me. I've enjoyed every moment because of the pleasure of conjuring up ideas out of very few facts. The next slide. Oh, I'm sorry. I forgot where here it is. Is a statement by Norbert Wiener. I've shown this slide before in lectures. It's important to me because Wiener is really the father of modern-day computers and communication techniques. He was really the philosopher that led to all of our ideas of how computers can be a new design of communication. But he had the recognition that, in fact, in general, there is no set of observations conceivable which can give us enough information about the past of a system to give us complete information as to its future. Meaning that, in fact, truth is always effervescent. You never really attain the final, complete bit of knowledge. You keep going. And that's also the pleasure of being a scientist, knowing there's always more. There's always more to come. And I think you'll see that in my lecture. Now, Dr. Fredholm made a few comments about me. I'm going to read now a bit about me. I was born in 1925. That was an interesting time, and for you students who didn't know about that time, it was a time in which there was no radio or TV. Actually, newspapers were the source of information, good information. And libraries were really the means by which we could attain books and read and so forth. But my father had a grocery store, and I lived on top of that store. I say this because he was a centerpiece for a large neighborhood. And I knew, as did my father, everyone in that community. We knew about them and we knew them. And for us, and for me particularly, retrospectively, I have felt the warmth of this community. It gave me the feeling about community and effort and society and what it means. And now looking upon my scientific community that made much of what I'm saying today possible. And here's a list of the people that worked in my laboratory over the years. I realized that my scientific neighborhood encompasses a place where cultural and language differences, because we did have many different languages, have been melded seamlessly and with synergy to promote communication. That, to me, was the essence of having a laboratory with these people. And to expand our knowledge with a kinship of purpose. We had a commonality. We knew what we wanted to do. And to create new thought. In this sense, nature, which we often equate with our genetic makeup, and nurture, which symbolizes our environment, interact mutually and synergistically in this community. These are the forces that have given meaning to life, and certainly mine. And that is the parable of which comes first, the chicken or the egg, is not of any biological importance. So my lecture symbolizes my interest in societal relationships. And it concerns the broad issues of biological communication. Most of my lecture will really deal with the past, but and how transducers and the concept of transducers came about. And since this concept is still at an evolutionary phase, I will conclude with an hypothesis which in its simplest message argues that biological communication consists of a complex meshwork of structures in which G proteins, the cytoskeleton, surface receptors, and the vast cytoskeletal network within cells are joined in a community of effort for which my life and those of my colleagues is a metaphor. So now I will begin the formal part of my lecture by paying tribute first, in this next slide, to what made me an endocrinologist. I was not trained as a pharmacologist or an endocrinologist. I came about it, and I'm not going to go into any detail, by this, what you're seeing here are fat cells, isolated from rat adipose tissue, which I'd learned how to do by using a collagenase preparation. And it was about that time, in 1963, to take you back that long, that a Nobel laureate, Bernardo Hussein, a great physiologist from Argentina, happened to be visiting my laboratory. And I showed him, under the microscope, these cells, and said, I've isolated them. And he said, but are they living? I said, what do you mean? I said, there's a nucleus. You can see that they have all the appearances of a fat cell. And he says, I won't be convinced unless you can demonstrate that they're living cells. And I particularly would like to know if insulin acts on these cells. I had no idea why he asked me that question at that time. But 2 days later, with the help of my colleagues Bob Scal and Sid Chernick, I put together the implementation to allow me to test the effects of insulin and discovered, yes, that insulin does stimulate by about tenfold the utilization of glucose. And I showed him the data about 3 days later, and he was ecstatic. He said, this is revolutionary. This will revolutionize everything in endocrinology. And to tell you the truth, I was not at all impressed. But it was because of his enthusiasm that I decided I would look up to see just what is this endocrine problem, what is insulin and all these hormones. So then I did begin to see that there is an interest. For one thing, it was not known at that time that insulin would act on an isolated cell. There was an idea that insulin would act on a tissue, a kind of propagated disturbance phenomenon. So this apparently gave insights in that insulin actually acts on cells. And then I tested the effects of phospholipases and proteases as a means of probing where the insulin receptor would be and where it propagates its action in a cell. And out of that came the knowledge that phospholipases would do everything that insulin did when you treated these cells. And so that was my first bold entrance into conjecture, and I said, well, insulin must be acting by activating phospholipases. Well, nobody paid any attention to that, fortunately. But then another event took place at the National Institutes of Health. I think it was in 1966, when Earl Sutherland came to NIH and gave a brilliant lecture about a new idea. This idea, which is represented here, is that on the surface of cells there exists receptors for a variety of what he called extracellular messengers in the form of hormones like adrenaline or peptide hormones or prostaglandins, each of which interact with receptors on the cell surface. And they, in turn, interact with an enzyme called adenyl, now called adenylate, and then adenylyl cyclase. All of that enzyme, in the presence of ATP, forms cyclic AMP, which he said is the second messenger. Now that discovery, which was actually the twin efforts of Earl Sutherland, and I'm happy to say Ted Rall, who I think is here today. He better be. There he is. Ted Rall really made most of the salient discoveries in this area. And was a pioneer in that, and I want to hail him for the efforts that he made back in the late 50s that made this discovery possible. The point is that this stimulated a lot of scientists at NIH to enter into this field because it became quite evident that if this is how hormones work, it should be possible to isolate these components and see how they interact and what happens. So then I had another person influence my career, namely a guy named Bob Williams, who is an endocrinologist at University of Washington at the time, who said, Martin, he said, look, I know you're a biochemist, but when you're dealing with hormone action, be very careful. Don't destroy everything in your process. Be gentle with your cells when you break them open and so forth. And that gave me the idea, okay, if I'm going to study this system, I'll study what I call fat cell ghosts, in which you just shock the cells and release the fat and have and the nucleus, and you would have then fat cell membranes. And then down the corridor was a man named Gopal Krishna, who had developed a very relatively simple test for the production of cyclic AMP by this enzyme. And he gave me that assay. And that was one important thing. And we then showed that, in fact, several different hormones would stimulate adenylate cyclase in these isolated ghost preparations. And then I had the great fortune to have come to my lab my first postdoctoral student, Lutz Birnbaumer, who with extraordinary prowess literally danced with this new method and produced a massive amount of data, which really, I think, was the promoter of all the ideas that we have about transduction. Now, what we effectively did was to study the actions of glucagon, thyroid-stimulating hormone, ACTH, luteinizing hormone, and epinephrine on the production of cyclic AMP. Now, Ted Roll had shown some years previously that, in fact, fluoride was able to replace hormones completely and would stimulate the production of cyclic AMP. Of course, we confirmed that. But in the process, we found that there was an important role of magnesium. Because if you just added magnesium to the system at high enough concentrations, you could replace the system with fluoride, or rather, you can replace fluoride or the hormones, and you would get full activity. But when you looked at the concentration dependency, we realized that we had at least two sites for magnesium. One involved with magnesium ATP, which is the catalytic site, and another which must be a regulatory site involved in fluoride action. And then we found that, in fact, ACTH, for example, would now decrease the concentration dependency for magnesium. At that point, we realized that we had some new clues about the regulatory process involved in the stimulation by hormones of cyclic AMP production. And then we proceeded with a variety of different indirect tests to demonstrate that, in fact, each of these hormones was acting through a separate receptor, as indicated by these different shaded areas of those little balls on the top of the slide. And then we were able to test something which necessitated having a homogeneous population of cells, namely the fat cell. Because now we could test, does each hormone act through a receptor that's coupled to its own cyclase, or are there separate receptors interacting with a common cyclase? And we did that by just simply adding, at varying concentrations, one or more of the hormones together, with the idea that if they were additive, that would give some idea that each receptor had its own cyclase. Well, we didn't find additivity. And from this came the idea of transduction, because then I realized that all of these receptors interacting with a common enzyme was something like having too many angels on the head of a pin. In other words, for me, an impossibility. And out of that came the idea of transducers. Now, I was fortunate in another way, because in 1969, I organized a meeting in tribute to Earl Sutherland. I invited him, plus scientists from all over the world who were interested in cyclic AMP and its metabolism. And the night before the meeting, a good friend of mine, who was actually competing with me on this same idea, named Oscar Hector, came to the dining room of the hotel where he was staying in Washington, and we sat for 3 or 4 hours that night thinking about all the different constructs that were possible. And he said, remember Norbert Wiener? He had some ideas about, abstract ideas about how you can deal with this process of transduction. And that led to this model. Using abstract terms, discriminator, which is a receptor, an amplifier, which is the enzyme, which now in its basal state or quiescent state doesn't produce much cyclic AMP, but which activated, it now produces a large amount of cyclic AMP. We call that amplification. And then a transducer, which somehow couples the information gained by the discriminator to the amplifier. And what we realized is that magnesium played some role in this, but we didn't know where. At that point, another person entered our lives, namely David Neville, who had just developed a procedure for purifying the plasma membrane from a rat liver. And we could get, therefore, a large source of membranes. David gave us that methodology. And then the other thing we liked about the liver is that we knew, had a receptor for glucagon, because that was one of the first receptors that were studied by Earl Sutherland and Ted Rall. And with having a polypeptide structure, we also recognized that we may be able to radiolabel this material, which we did, and then study the binding of the hormone to the discriminator and try to relate that binding event to the amplification event. And so Michiel Krauns came to the lab, and I assigned to him the role of studying the binding of radiolabeled glucagon to the receptor for glucagon. And what he had found was, and he was very exasperated by this, was that it took a long time for the binding to occur. That is, it would take till steady state occurred maybe 20 minutes. Whereas we already knew from studies we had done with that system that glucagon initiated very rapidly, within seconds, the production of cyclic AMP. And if you added an inhibitory form of glucagon, which was des-histidine glucagon, which was given to us by Sundby in Denmark, then we found that the system would decay very rapidly. So we expected the binding to show the same characteristics. Instead, they showed slow binding and effectively no release of the hormone once it was bound. But then we realized that we were using a very simple medium for studying the binding. And so when we went to the cyclase medium, that is the medium we use for measuring cyclic AMP production, we had to, of course, add magnesium and ATP and other substances. And then we found a dramatic change in the binding. It reached steady state very rapidly, and it rapidly decayed if we added des-histidine glucagon. So in finding out what is the culprit that caused all this, we realized that ATP was the one factor that was changing the binding the way it did. But when I was a graduate student, I made the mistake of thinking that everything was pure when you bought it from a pharmaceutical company. And as a result, I made a fundamental error in my thesis by saying that ATP was the source or the substrate for phospholipid synthesis. It turns out that it was CTP, which is a contaminant of ATP. So I immediately went down and gathered as many different nucleotides that I could. And to make a long story short, of course, it turned out that GTP at very much lower concentrations would do everything that ATP did. And at that point, we said to ourselves, now we should see, is GTP involved in hormone action? Now you realize that ATP is the substrate for the enzyme. So I went down to Marshall Nuremberg's freezer, which is down the hall, and stole from his rack some enzymatic components that were necessary to make a derivative of ATP called AMP-PNP. That derivative is not hydrolyzed by ATPases, but more importantly, when I made this material, it was no longer contaminated with GTP. And when we then studied in the upper left panel the effect of hormone, we found effectively the hormone would do nothing if you left out GTP. If you added GTP to the medium, you got some stimulation, but now you got a very large increase in cyclic AMP production when you added GTP plus hormone. At that point, we realized that we had a key to what transduction might represent. Namely, there was some structural component of the system which require GTP for hormone action. We also ultimately turned to the fat cell, and many other systems showed the same thing, in which as you vary the GTP concentration, you would first get stimulation, and then you'd get inhibition. And then entered a big Swede, a good friend of mine, Hans Löw, and Jim Harwood, who then found out that fluoride would do the same thing. Fluoride would stimulate at relatively low concentrations, and at higher concentration, it would inhibit. That would happen in some systems, but not all systems. And then we realized that the system must be much more complex than just having one type of process. There must be another inhibitory process. And later on, Dermot Cooper and Dean Londos discovered that, for example, fat cells contain adenosine receptors, which selectively cause the inhibition of adenyl cyclase activity by a GTP-dependent process. That was already established by my first Japanese colleague, Hirohei Yamamuro, who was even a better dancer with cyclase activity than was Lutz Birnbaumer, and he would produce an enormous amount of data which convinced me that, in fact, we were dealing with two separate processes, one causing stimulation and the other inhibition. And we call these NS for stimulatory and I for inhibitory. They have become now GS and GI for stimulatory and inhibitory. These are the G proteins. Well, at this point, we realized, as I said, that we had a much more complicated kind of system involving two independent processes. And we drew then this diagram saying that GTP must be reacting with an independent structure or structures, which was now changing the dynamics of both the discriminator in its binding and the cyclase in its activity. And that became, to us at least, the forerunner of the whole concept of transduction. And then Yoram Solomon in my lab said, you know, Marty, you made AMP-PNP. Why don't we see what would happen if we made GMP-PNP, a non-hydrolyzable analog? Then we don't have to worry about GTPases, and we can see what happens. Well, it turned out that GMP-PNP stimulated the enzyme in the absence of hormone by a time-related process, and in such a fashion that it effectively caused the enzyme to become irreversibly activated. And that surprised us. But it gave us the first clue that there was another dynamic process going on, namely the hydrolysis of GTP may be part of the dynamics. And so Mona Sperman and Mark Rendell decided they would model the kinetics, the effects of magnesium and GPP and HP on these systems, and they came up with this three-state model in which one goes from E to E prime, which doesn't give any increase in activity, but then it switches over into a new state, which now has high activity. And if you took the same data, but instead of GPP and HP, put GTP in there, you got a whole new phenomenon, because now you got a process in which the system was turning over rapidly, and you could explain all of the dynamics of how hormones rapidly initiate the activity and keep it going as long as the hormone is there. The GTPase aspect of it was very important. And during that time, Cassell and Selinger, in a brilliant set of experiments, came up with the evidence that indeed there is a GTPase that is hormone stimulated, and they furthermore showed that cholera toxin, which was already known to affect cyclic AMP production, would work by inhibiting the GTPase activity, therefore keeping it going. And I've just represented their diagram of how this may occur. Out of their modeling, actually, came the concept that what the hormones may be doing, or are doing, is increasing the exchange of bound GDP, which is an inhibitor, with active GTP, the activator. And that's how hormones are working. And I think this has been confirmed by a large number of workers in the field. But there was one thing that bothered us about all of this, and namely, Ann Welton and Pramod Lad and Simonetta Nicosia, in the meantime, were studying the extraction of the membranes from liver, for example, and trying to see if they can separate the N protein, we call, or G protein, attached to the receptor from the N or G protein attached to cyclase, to see if we could separate them and understand their characteristics. Well, we found by a variety of techniques that, in fact, when G is attached to the receptor, it has very different properties from the G factor when it's interacted with cyclase. For example, GTP would be necessary to stimulate cyclase. GDP would be an inhibitor. Whereas the effects of these two nucleotides on hormone binding were identical. So therefore we knew that that's just one example, and I've given others here, which suggested that somehow the receptor, when it's interacted with G, was communicating with the enzyme in a fashion which would change the structure of G from one form to another form. And that led to what I consider in our lab the revolution in how we have thought, and I'll now describe in very briefly the contemporary way that we are thinking. And this came about, and you'll think this cartoon is silly, I know, by using a tool called irradiation analysis, in which you shine a beam of very high energy electrons at, for example, liver membranes. And you follow the activity of a system, such as adenylate cyclase, to see what would be the effect on radiation on the decay and activity. The radiation, I should tell you, is such that it kicks the backbone of the proteins in these systems so that they no longer show activity. We now know they don't break the molecules apart. They just fragment them to the point where no more activity is exhibited. So if you follow the rate at which the system decays versus the amount of energy that is put into the system, one can achieve some estimate of the functional mass. And this cartoon is just to illustrate that if you have a series of balloons and you're firing at them, the greater chance of hitting one of these balloons is when it's large. And you consider the volume and you consider the mass, then the rate at which this bursts is going to give you some idea of the functional size of the system you're looking at. And this allowed us for the first time to look at what is happening when the membrane is intact. You don't have to extract anything, just to see what's happening within the membrane environment itself. And so out of this came a rather surprising result. Namely that the system, the adenylate cyclase system in rat liver membranes, before you touched it with GPP-NHP or GTP or hormone or fluoride, had a very large size, over 1 million Daltons, which was much larger than anything we knew about these systems. We knew, for example, from Al Gilman's work and from Pfeiffer, that the size of what we called at that time the GTP component was something like 45,000 in molecular weight, whereas we were getting something considerably larger. And then the most interesting thing happened. If we now expose the membranes first to GPP-NHP or fluoride, or to glucagon plus GTP, there was a big reduction in size, indicating that something was converted from a large size to a much smaller size. Then when we looked at what we call the RN, or the RG complex, we found that it too was much larger than what we could recognize from the size of the components themselves, the receptor and the G protein. And from that, I published a model. I was always publishing models, which suggested that perhaps the system exists in some kind of aggregated state in which you had the receptors and the G protein, this is the black and the white areas in these circles, coalesced in some kind of oligomeric structure, and that the hormone and the GTP together in some concerted fashion were disaggregating this structure, giving rise to something which now had a receptor and a G protein, but in a smaller, it's just one of these subunits, in other words, of this multimer. And that is the active material which is relayed to C, the catalytic unit, making it now active. So that gave us the first clue that perhaps we're dealing with structural changes in the G protein, giving rise to the new form of the G protein attached to the enzyme. Two assumptions I had made at that time. One is that the G proteins and the receptors were in some stoichiometric relationship. And actually, that's all we could think about at the time. Now we know, years later, that in fact that's not true. There are many more GS, for example, than there are receptors, or for that matter, other G proteins. So that couldn't be in the style that I'd drawn here. In other words, there are more GSs than there are receptors. The other thing is we didn't realize at the time that there was a heterotrimeric structure. That is, that G proteins are composed of alpha, beta, gamma. And that came about by a consortium of a number of studies. Now, Gilman will discuss this as how this came about. But the important thing is, for us, is that the knowledge that you had a heterotrimeric structure, alpha combined with a beta-gamma complex, that when occupied by GTP, is represented here, that you would split these two fragments, A, alpha from beta-gamma, producing an alpha with a GTP on it and a beta-gamma. And now we know that they operate distinctively on different systems, which again, Dr. Gilman will talk about. So everybody forgot about my model, because this was a much simpler thing. You start with a heterotrimer, you split it up, and the individual components do everything. But I became convinced that we were dealing with something more complex. Remember, think simplicity and throw it away? Well, that's what I did. And so, unfortunately for me, I became an administrator for about several years. But then when I did go back into doing real science, I realized that we had to follow up our studies. And I'm not going to show you data, but I'm going to just give you one evidence for a multimeric model of hormone and GTP action is the use of cross-linkers. That is, if you cross-link a polymeric structure, you should be able to isolate this structure. And effectively, we showed that all of the G proteins, the heterotrimeric G proteins, in intact membranes could be easily cross-linked. The only two kinds of proteins that could be cross-linked in isolated membranes or associated with these membranes were actin, which is known to form polymers, and tubulin. And I don't think I'll show you these other data. This is data describing that. This is control forming one peak of material after you've before cross-linking, and after you cross-link, you've got very large structures, much larger than the control. And from this, we realized that we were really dealing with multimeric structures of G proteins. And I'm going quickly because I have to. But then we went through a series of studies and properties of G proteins extracted with a detergent called octoglucoside, sedimentation analysis, effects of hormones and guanine nucleotides on sedimentation, cross-linking, and then the effects of a variety of different detergents to try to extract these things. To make a long story short, and all of this has been published, we realized that we may have something that's something similar to the structure of cytoskeletal proteins. And I represent here what the dynamic instability model for actin and its dissociation into individual monomers. I say giving this slide because it will demonstrate how I'm thinking today of how G proteins operate. In effect, a filamental structure of actin, which is part of the cytoskeleton of cells, and in fact is essential in muscle for muscle contraction, that this structure has an asymmetry in which if you have ATP bound to monomers on one end by the color orange, and ADP bound on the other end, represented by the color green, that you set up a dynamic instability in which now a monomer will come off with ADP on it from this end, and on this end, one containing ATP will go on that end. This was a powerful stimulant for me to think of how you could have a receptor attached to multimeric G proteins and get them to function by this kind of array of disassembly and assembly, depending upon whether you have occupied with GTP or ATP or ADP. And then the other thing that I found was that when thinking of muscle contraction, you have two types of proteins. You have the actin filament, this large multimeric structure, to which is bound myosin. The myosin molecule rides along this filament, much like a train on a railroad track. And in doing so, it's propelled by the exchange of ATP with ADP at each step. And that helps the propulsion. The other thing that happens is that ATP is hydrolyzed in this process to give rise to ADP plus PI. And that when the PI, or the phosphate, inorganic phosphate, is released, now you revert back to the original state. So this was the dynamics and the control by ATPase of the movement of myosin along this chain, which allows us to pull and tug and play tennis and all those things. And so from that came this simple version that what we may have for signal transduction with G proteins is something similar. In the sense you have a receptor embedded in the membrane, which is somehow attached to this filamentous form or multimeric form of G proteins containing either GTP, represented by T, or GDP, represented by D. And that as the exchange is occurring between GTP and GDP, as the receptor rides along its rails, you're eventually going to saturate one or more of these substances with either GTP or GDP, and you'll get a fall-off on one end of active GDP and recoupling of inactive or GDP form coming back on the other end. Now, the reason that all of this made sense to me, you'll see in a moment, and again, this is a slide that I'm sure that Al Gilman is going to talk about, namely, now that we know from his work that adenylate cyclase, which is the ultimate recipient of all of this business, is really a molecule which is spanning the membrane 12 times and is hooked together by one peptide linker and forms essentially two domains, which I just listed here as M1 and M2. Now, the interesting thing about this structure is that depending upon the isoform of adenylate cyclase, and there are many more than the 5 listed here, beta gammas can serve as the inhibitor of cyclase. It can serve in one form, the olfactory, as a stimulatory, whereas alpha S, of course, is always an activator. Beta gamma can sometimes synergize with it and increase it. So you can have different forms of cyclase now exhibiting different behavior of the alpha subunit and the beta gamma subunit. And out of this came the following model, and I hope it's not too complex for you to follow. I'll just lead you through it. And that is, here is the multimer attached to the receptor. The hormone reacts with the receptor and causes the exchange of bound GDP with GTP. And now the receptor is moving along this, catalyzing each time the exchange reaction, until finally, due to the asymmetry of this structure, GDP comes off as the monomer. This is the heterotrimer monomer with the GTP bound. And now it interacts with this enzyme, which, as I say, is in this M1, M2 structural form, and forms E star, which is equivalent to what we had modeled as E prime, which is not active, but which now contains this. Then the crucial thing is magnesium. Here, magnesium comes and causes the splitting of the GTP to GTP-PI, or inorganic phosphate, which are still bound to the molecule, the GTP alpha subunit, whereas the beta-gamma segregates onto another component, namely this is would M2, for example. And out of this activated state, the E double star, comes the production of cyclic AMP. The rate at which this system decays is related to the release of the bound inorganic phosphate from the structure, giving rise to, again, the monomer with containing bound GDP, which can go back and recycle. So this whole system now consists of two cycling processes. One which relates to what is happening to the G proteins themselves, influenced by the catalytic action of hormones, and the other which is influenced by the role of magnesium and GTP and the GTPase of this combined structure, giving rise to the dynamics. Now, enough of that kind of model. What I think now for the future is as follows. There's an increasing amount of evidence that not only do we have multimeric G proteins, cytoskeletal in nature, but we have some interaction between the cytoskeletal network within the cell with these multimeric G proteins. I'm glad to hear that Eva Sarndal is here, because she's done some important work in that area, demonstrating, in fact, that there must be some kind of association. And out of this association, I believe, is going to be a key in future research to really what cell signaling is in the intact cell. And I just represented this by my last slide, which demonstrates in an abstract sense that where we are going, I believe, is that the entire meshwork of structures within the cell, the cytoskeletal network, the G proteins, are all combined with various types of receptors on the cell surface to give rise not only to chemical signals like cyclic AMP or ion channel changes, but also in the ability of cells to move and change their shape, which is really the hallmark of the action of many hormones on cells. So in this sense, communications is really the result of many different interactions. And this really represents, as I said when I read the opening part of my lecture, a community effort, an effort by all components of the cell to bring about the entire change in the physiological basis or structure of the cell in response to hormones. And with that, I end my lecture. Thank you.